All right, welcome to Chem 313, Lecture 17. Today, we are going to finish up with the biological energetics or metabolism section of Chem 313. And we're going to cover a lot of ground today. So one thing that we're going to talk about is the pathway of gluconeogenesis, which is synthesis of glucose. And we're also going to talk about hormonal regulation of metabolism. So we're going to talk about a lot of material today, but the amount of depth that you're expected to know it in is pretty shallow. I'm not going to expect a complete recounting of every single aspect of these pathways. So, once again, you want to know the usual stuff as far as gluconeogenesis is concerned. That is, where and when, the point or the goal, the major reactants and products, which is a bit complicated for gluconeogenesis because there are several possible reactants, and lastly, regulation. As for the hormonal regulation of metabolism section, simply know the effects and the logic of why these hormones do what, which is fairly intuitive. Okay, let's talk about gluconeogenesis. Let's break the word down. Gluco means glucose, neo means new, and genesis means synthesizing things, right? So this is synthesis of new glucose, okay, from scratch. So in a sense, gluconeogenesis is just glycolysis run in reverse, although it's a little bit different for reasons that we're going to see. All right, so where and when does this take place? Well, it takes place in the cytoplasm, and in animals, it mostly takes place in liver cells. Some other tissues can do gluconeogenesis as well, but for animals, it's mostly the liver. The liver is what's responsible for making all of the glucose that you make yourself. When is going to be when ATP is abundant, and carbon sources are available. We'll talk about that in a moment. All right, the purpose or the goal of gluconeogenesis is, well, we're trying to make glucose, but what's glucose used for? So glucose is a fuel source, right? We're going to use it eventually to do glycolysis and get some ATP. But typically, when gluconeogenesis is running, we don't need glucose right away, at least not in the liver. So typically, when the liver is doing gluconeogenesis, it's going to do one of two things with the glucose. Either secrete it into the blood, and usually what that will do is it will go and it will feed muscle cells or brain cells, which are all very big ATP consumers, or it will store the glucose as glycogen. We talked about glycogen earlier. Glycogen is that helical glucose polymer that's used as a storage form of glucose. And if the liver makes enough glucose and the cells aren't using it up right away, the liver can store it in the form of glycogen. Muscles can also take in glucose and store it as glycogen. All right. As for the regulation, we will talk about that on the next slide. And as for reactants and products, well, it's tricky. So there are several possible raw materials for glucose synthesis. All right, this is an anabolic pathway, and one of the features of anabolic pathways is they tend to diverge a lot compared to catabolic pathways. So before I tell you what the major reactants and products are that I expect you to know for gluconeogenesis, we need to set up some comparisons between gluconeogenesis and glycolysis. So let's take a look at that. Gluconeogenesis is almost glycolysis in reverse. So let's take a look at glycolysis. We have 10 reactions. We start with glucose. And I'm using a one-directional arrow for reversible reactions and equilibrium arrows for reversible reactions. All right, so notice that we have 3 irreversible reactions of glycolysis, and those are your 3 regulated steps. Steps 1, 3, and 10. All right. Now, those steps are irreversible. The reason they're irreversible is because the free energy change for those reactions is extremely negative, which means that if we were to reverse these reactions, the delta G would be extremely positive, okay? So that means gluconeogenesis cannot simply be glycolysis in reverse. Gluconeogenesis must undergo some sort of alternate reaction to bypass the 3 irreversible steps. All right, so how does it do that? Well, there are alternate reactions. And you've probably guessed that ATP is going to be involved, and you would guess correctly. So to answer the question, what are our major reactants and products of gluconeogenesis? Well, we can simply start from pyruvate, but as it turns out, if we want to reverse the process of gluconeogenesis, we can't simply run reaction 10 in reverse. What happens is we combine pyruvate with CO2 to give us oxaloacetate. And this is an ATP-dependent step. And then oxaloacetate can be made into PEP, which is the product, or excuse me, the reactant of reaction 10 of glycolysis. So that's one of our bypass reactions, and this process requires ATP. Now, the reason reactions 1 and 3 are irreversible specifically is because these are the 2 steps that involve ATP hydrolysis in glycolysis, and we know that ATP hydrolysis is very favorable. So if we were to run these two reactions in reverse, we would be synthesizing ATP, which is very energetically unfavorable, right? That's why we had to do all this oxidative phosphorylation in order to power the synthesis of ATP. So what ends up happening at these steps is that we do not have ATP synthesis in glycolysis, in gluconeogenesis. We simply have hydrolysis reactions instead, not involving ATP. And you don't need to know what these reactions are, so don't worry about it. Okay? The point of mentioning this is because I want you guys to understand that if we simply ran glycolysis in reverse, glycolysis gets us 2 ATP, right? So if we simply ran all the reactions in reverse, we would consume 2 ATP. But in reality, it's not that simple. We have to bypass these steps using hydrolysis reactions. And so therefore, the reactants and products of gluconeogenesis and glycolysis are going to be different. So if we start our gluconeogenesis from pyruvate, these are our major reactants and products. 2-pyruvate plus 2-NADH plus 6 ATP react to give us glucose plus 6 ADP plus 2 NAD plus. That's assuming that we're starting from pyruvate. So notice it takes more energy to make glucose than is recovered in glycolysis. Right? Glycolysis gets us 2 ATP, but it takes 6 ATP to do gluconeogenesis. And why is that? Well, that's because we have to do our bypass reactions. Because glycolysis is so favorable, we have to put energy into our system if we want to run it in reverse, using alternate reactions with ATP hydrolysis. All right, so this diagram shows the flow chart of how gluconeogenesis actually works in liver cells. We can do gluconeogenesis starting from pyruvate if the liver has plenty of pyruvate on hand, okay? However, what I've done is highlight in blue the different substrates for gluconeogenesis. By substrates, I mean simply raw materials. So pyruvate is only one raw material that we can use to make glucose. We can also start from amino acids. So why would amino acids be useful to make glucose? Well, remember, the whole point of making glucose is that you eventually want to secrete it and use it as an energy source later on, right? Which means that we need an energy source. Well, when will free amino acids be floating around that we can use to make our energy molecules from? Well, this is an important source during starvation. Because if you're starving, your muscle tissue is going to be broken down. And one of the important things that can be done with the muscle proteins is to chop them up into amino acids, which can then be made into pyruvate or other products like alpha-ketoglutarate, and then eventually used to make glucose. Another important raw material for gluconeogenesis is lactate. So lactate can come from the muscles, and this is usually done during heavy exercise. Why is that? Well, that's because during heavy exercise, when you're not breathing enough to keep up with the ATP demand, you're going to form a lot of lactate because the muscles are going to be trying to get as much ATP as fast as possible by using glycolysis only and forming lactate as a byproduct. So what ends up happening is that the muscles secrete lactate into the blood. It travels to the liver, where it's taken into the cells, converted into pyruvate, and then used to make glucose, which can then be sent back to the muscles. Another important source of glucose is actually glycerol. You're not responsible for knowing that, but that's also an important source of glucose during starvation because you're going to be breaking down your adipose tissue. Adipose tissue contains triacylglycerol. We've already seen what happens to the fatty acids. They can be broken down by beta oxidation. The glycerol can be broken down and made into glucose. And then there are various, in purple, products of gluconeogenesis. We already said before that the two major products of gluconeogenesis are glucose, which is for immediate use, and by that I mean secretion into the blood so that your brain and muscles can use it right away. And then the other major product is glycogen, which is storage for later use. All right, so the core pathway of gluconeogenesis is shown on the diagonal through here. You are not responsible for every reaction. It's simply the reactions of glycolysis run in reverse with bypasses on steps 1, 3, and 10. All right, let's take a look at the regulation of gluconeogenesis. AMP inhibits gluconeogenesis and ATP activates it. Notice that this is the exact inverse of glycolysis, right? Glycolysis is inhibited by ATP and activated by ADP and AMP. So notice that gluconeogenesis is activated by a high energy charge. We saw that term before. The energy charge you can think of as the ratio of ATP to ADP and AMP. And so if you've got a lot of ATP lying around because you're not using it, one thing the liver can do with it is to put it to good use by making glucose. All right, so this is reaction 3 of glycolysis. So this would be reaction 8 of gluconeogenesis, that step right there. It's also inhibited by fructose 2,6-bisphosphate. We've seen that molecule before as well. Remember, fructose 2,6-bisphosphate activates reaction 3 of glycolysis. It inhibits reaction 8 of gluconeogenesis. So fructose 2,6-bisphosphate activates glycolysis and inhibits gluconeogenesis. This is what's called reciprocal regulation. Why is that? Well, fructose 2,6-bisphosphate is made from glucose. So if you have a lot of fructose 2,6-bisphosphate in your cell, it must mean that you've got a lot of glucose lying around. If you've got a lot of glucose lying around, you do not need to be wasting ATP making more glucose. So that's the logic behind this reciprocal regulation. Glucose 6-phosphate also inhibits. Glucose 6-phosphate inhibits reaction 8. And so this one reaction is our primary control point for gluconeogenesis. All right, so let's take a look at how we regulate all these different pathways. They seem to almost be competing with each other, and they are. So for example, liver cells are not going to want to be doing glycolysis and gluconeogenesis at the same time. That would be complete chaos, right? So cells, and especially the liver and other cells that undergo a lot of metabolic reactions, they need some way of knowing which reactions will run at a given point in time. So we're going to take a look at the hormonal control of metabolism really briefly. Let's take a look at the short-term regulation of metabolism first. By short-term, I mean minutes to hours. All right, so we're not talking the time scale of your life. We're talking about the time scale of this afternoon. So let's take a look at what happens during a well-fed state. So this is when blood glucose levels are going to be high. So this is the situation that happens after you have a meal. So what happens when blood glucose levels rise is the pancreas secretes insulin. We saw insulin in the cell signaling portion of the course, lecture 11. So insulin, you'll recall, is a hormone that sends a message to cells. It says, take in glucose, use it, and don't use other fuels. Okay? The reason insulin tells cells to take in glucose and use it is because glucose is not something you want a whole lot of floating around in your blood for long periods of time. It puts osmotic stress on your cells. It has to be filtered out by the kidneys. Having persistently high blood glucose levels is a dangerous thing. So insulin, its goal is to tell cells to take in glucose, use it for whatever, and don't use other fuels. So what do you suppose are the pathways that insulin is going to tend to activate? Well, what's going to happen is in the liver, glucose will be taken in from the outside. Then what it's going to do is run glycolysis to make pyruvate and acetyl-CoA. And that can be used to generate ATP. Now, we may be in a situation when the liver doesn't need ATP. Well, that's fine. Some of that glucose can be stored in the form of glycogen. The rest of the glucose will be picked up by other tissues such as the brain and the muscles and used to make ATP. Now, if you eat a lot of carbohydrate, if you eat a lot of sugar, there's only an upper limit to how much carbohydrate you could use at one time. So any excess carbon from that glucose will be converted into acetyl-CoA, and if your ATP levels are high, which means that you're resting or you've just got an abundance of energy, you don't need to be doing the citric acid cycle. So instead of the citric acid cycle, what's going to happen is the acetyl-CoA will be made into triacylglycerols and then stored in adipose tissue. So to summarize, the effects of insulin signaling, and again, this is short term. We have glucose absorption, that's the take-in glucose part. We have oxidation of glucose, that's the use it part, okay? And so this is going to work until ATP stores are abundant. At that point, we don't need any more ATP in our liver, so what's going to happen? Synthesis of glycogen. And that works until glycogen stores are full. And any leftover after that we'll make into acetyl coenzyme A, used to make fatty acids. And then the fatty acids will be stored in the form of triacylglycerol in the adipose tissue. All right, once again, short term, let's look at the inverse situation. Starvation or stress. We're talking about either situations of low blood levels of glucose or high demand for glucose, which is what happens during stress. All right. In this situation, the liver produces the pancreas produces a different hormone called glucagon. I know that terminology is confusing because we got all these words that start with G and have 3 syllables, but bear with me. All right, so glucagon is a separate hormone. It tells cells to release glucose and use alternate fuels. Why use alternate fuels? Well, because we're in a situation of low glucose, and so if cells are going to be dependent on glucose as an energy source, they're going to be coming up short very quickly. So what does glucagon do? Well, primarily in the liver, at least, we have to release glucose. How do we do that? By promoting gluconeogenesis. So glucagon activates gluconeogenesis. It also activates breakdown of glycogen. Why? Because we're in a low glucose stress. We have to take all the glucose that we have, such as glycogen, and release it so that other tissues can use it. We then secrete glucose in the bloodstream so that it can feed the brain. This is important because the brain can really only use glucose as a fuel source, glucose and something else called ketone bodies. All right? Now, as for the use alternate fuels part, we have to go to the adipose tissue. So remember, when we were talking about beta oxidation and the use of fatty acids as fuels, there was that enzyme called hormone-sensitive lipase. Hormone-sensitive lipase is activated by glucagon, which means that fatty acids are going to be released into the bloodstream. So what happens to our fatty acids? Well, they can be sent directly to muscle to be used as an energy source. So that's the use alternate fuels part. They can also go back to the liver, where they are broken down into molecules called ketone bodies, which can be used to feed the brain. So we haven't talked about ketone bodies in this course, and you're not responsible for knowing what they are, but they're a byproduct of glucagon signaling. Additionally, and we didn't talk about amino acids as an energy source in this course, but glucagon also activates protein breakdown to form pyruvate so that we can run gluconeogenesis. So to summarize, we have to use alternate fuels. So fatty acids are absorbed, I should say, by liver and muscle, and are broken down. Glycogen is broken down, gluconeogenesis is activated. Okay, let's take a look at the longer-term regulation of metabolism. So longer term, we are talking about on a time scale of weeks to months. So if you study biology for any length of time, you learn that there are a whole bunch of overlapping signaling pathways that sort of nest within one another. And there are different hormones that we don't have time to talk about their specific pathways, but we can talk about their effects. All right? And so one of those important hormones is called leptin. So what is leptin? Leptin is secreted by adipose tissue as it grows. So the more adipose tissue that you have, the more leptin that you have. Leptin is intended to inhibit food intake and activate fatty acid breakdown. Right? It's secreted by adipose tissue. The more adipose tissue that you have, leptin will both decrease intake of food and also decrease the synthesis of new fat, and it will activate beta oxidation pathways to break down the fat that you have. So this is a way of feedback regulation of forming new adipose tissue. It's a way of preventing adipose tissue growth from getting out of control. The more of it that you have, ideally, the more beta oxidation of fatty acids that you'll do and the less food you'll take in. So needless to say that decrease in leptin secretion is a very important contributing factor in obesity. In fact, it can be shown that mice that are genetically altered so that they cannot produce leptin, they become morbidly obese even on a regular diet because their fat synthesis is activated and they're not breaking down any of the fatty acid that they have. And then sort of the inverse situation to that is a hormone known as adiponectin. So adiponectin is produced by adipose tissue, but only in response to shrinkage of adipose cells. So in other words, when you're losing weight. So adiponectin is produced by shrinking adipose cells, and it's secreted and it goes to several different tissues, and the most important target is this molecule called AMPK. AMPK stands for AMP activated protein kinase. So just like protein kinase A that we saw with the epinephrine signaling pathway, AMPK is a protein kinase. It phosphorylates other proteins, turns them on or off. All right, so if we have adipose cells that are shrinking, what does that mean is happening? Well, it could be several things, but what your body thinks is happening is that you're starving. And so if you're starving, what are the goals? Well, the goals are we need to release fuel sources from our stores, and we need to conserve what fuels we have. All right. So what does AMPK do? Well, for example, it does an awful lot of things, but for example, it promotes fatty acid beta oxidation and inhibits fatty acid synthesis. Does that make sense? Well, yeah, if we're starving, we need to use fatty acids as an energy source because we don't have a whole lot of glucose on hand. Okay? However, somewhat contrary to what you might expect, it inhibits breakdown of triacylglycerols. This is one of the reasons why if you have already lost a lot of weight, it gets harder and harder to lose more weight as you go. Okay? We also, however, have an inhibition of lipid synthesis because once again, we want to conserve our fuels. Okay? Furthermore, we have an increase of glucose and fatty acid uptake. We have an increase in feeding behavior and a decrease in energy expenditure. And we have a decrease in insulin secretion because insulin basically undoes everything that adiponectin is trying to do. All right? So basically what we do with AMP kinase is we activate catabolism and inhibit anabolism. So activating AMP kinase is an important therapy in metabolic disorders such as obesity and diabetes. One of the most important drugs out there for treatment of diabetes type 2 is metformin. Metformin activates AMP kinase. It does an awful lot of other stuff as well, but that's one of its most important functions. All right, so what's the point of all this? So the point of all this regulation of metabolism is to ensure survival by making wise use of energy sources. Okay? If we are in a situation where energy is abundant, okay, and that means that in cells, ATP levels are high. If your ATP stores are high, there's no point in doing catabolism. So we do anabolic processes. So for example, gluconeogenesis or fatty acid synthesis, which we haven't talked about. If we're in a situation where fuels are abundant, which is not necessarily the same thing as a situation when energy is abundant, so that means we have high glucose and or fatty acids, we need to take in those fuels, use them if possible, and if that's not possible, store them. If we are in an energy limited situation, so that means inside the cells you have low ATP, we need to do catabolism. So that could mean glycolysis, the citric acid cycle, beta oxidation. And if we're in a fuel limited situation, so that means low glucose or whatever, then we need to release fuel stores and conserve. All of this stuff, I think, is quite intuitive. And this is about the level of detail that I expect you to understand it. So go back and review gluconeogenesis. I know it's more complicated than glycolysis, but there's less to know about it. And then go back and just briefly overview the take-home points from each of these slides on hormonal regulation. And you can expect to see this material on quiz 4 and on the final exam. We're now done with the bioenergetics section of the course, and for the final few lectures, we are going to talk about biological information, and that involves introducing our final class of biological macromolecules, nucleic acids. So you have that to look forward to in the following lecture, and I will see you at office hours.